Hi class, and welcome to your next video on cellular transport. Now we've talked all about the organelles of the cell and how they work in combination together. Now we want to talk about one of the ways we use energy, which is to transport things in and out of the cell. And one of the main components of cellular transport is the plasma membrane. And we've talked about the plasma membrane and thus that it makes up the boundary of the cell, but we want to talk about it even more in how does it help with cellular transport. So we're going to focus on that, and we're going to focus on how cellular transport occurs. So this is what the plasma membrane looks like up close. And as you can see, we have a lot of components here. Here is the outside of the cell, and here is the inside of the cell. This is where the cytoplasm and the cytoskeleton that gives us a lot of structure and rigidity. But if we look here, we have those phospholipids that we talked about in chemistry of life. Remember that the phospholipid is made up of a phosphate and two fatty acid tails. And that's the main component of our plasma membrane. Now we have a few other things. We have proteins, which are going to help things come in and out of the cell. We have cholesterol, which is going to help the whole plasma membrane with its structure. And we have some carbohydrates over here, which are going to be kind of like flags saying, hey, I'm a bone cell, hey, I'm a plant cell, hey, I'm a skin cell, so that we know exactly what kind of cell this is that we're dealing with, because when cells talk to each other, they need to know what each other is. So when we talk about the plasma membrane, we talk about the fluid mosaic model. Now what that means is everything in the plasma membrane is movable. Everything is not locked into place, and everything can move around and change places based on how molecules are crossing that membrane. So nothing is stuck in place. Everything can move. If we need to get a carbohydrate across and it's really big, we might move some of our proteins to get it to the right spot to let that carbohydrate across. So the plasma membrane consists of two layers of phospholipids, and we call this a bilayer. Now if we go back here, you can see that there is one head sticking up towards the outside of the cell with the two fatty acids, and one head sticking down towards the inside with two fatty acids sticking up. And that's what makes up our bilayer. Now the proteins that help with transport are kind of just embedded all along. As we can see here, there's a protein here, a protein here. So they're kind of just embedded everywhere all along. And those are going to help with our transport. They're going to be pumps. They're going to serve a few different roles within our plasma membrane. Now the cholesterol, like I said, is for rigidity and support, and it helps just give us some support in that plasma membrane, because remember, everything's movable. We need a little bit of rigidity. We need a little bit of support. And then finally the carbohydrates, which are in the extracellular matrix, they're outside of the cell, help identify the cell for any hormones, anything coming in. Now, let's talk specifically about each part. So the phospholipid, if you remember, is made of a phosphate head, which is polar, and it is hydrophilic, meaning it is water-loving. So water can cross this phospholipid bilayer, but it doesn't cross it very easily, because then it gets to the fatty acid tails. And the fatty acid tails are hydrophobic, meaning they don't like water. So it becomes very difficult for water to cross, even though it can cross. Then we have the proteins. Now we've talked about the proteins being pumps or being there for transport. And the proteins are embedded in the membrane function to support and transport substances into and out of the cell. Some are channels and some are pumps. So if we see here, they might just let something through. They might attach somewhere and then open or close. So there are all kinds of different ways the proteins can interact. And the proteins are really there to just help things across that membrane that can't get across the phospholipid bilayer, because there are some things that might be too large, that might be charged, that cannot cross that phospholipid bilayer. So we talked about cholesterol, and cholesterol functions to provide support for making the membrane and making it more rigid and stronger. Now, think about our chemistry of life unit. Do you remember what this was? If you said a steroid, you would be right. And look at the structure. It is a ring. And that ring is going to help us with the rigidity and support that we need. And lastly, we have carbohydrate chains. These chains are made up of monosaccharides to help identify the cell. So whenever a hormone or a steroid recognizes a cell, these are kind of like a flag. Like a country has a flag, well, so do cells. And they're saying, hey, I am whatever kind of cell. So our whole plasma membrane is selectively permeable. What that means is some things can pass while other things have difficulty. They're kind of like a gatekeeper or a bouncer at a club. If your name is on the list, you're getting in. If it is not, you are not getting in. And that is how the plasma membrane works. If you have certain properties, you can cross the plasma membrane very easily. You get by on that lipid bilayer very easily. If your name is not on that list, you have a whole different way you have to go through, and it becomes very difficult. So the things that pass through easily, though, are going to be small molecules, nonpolar molecules, for example, oxygen, carbon dioxide, or water. The things that might need some sort of assistance are going to be very large, charged, or polar molecules, for example, glucose ions, and even your vitamins. So we've talked about what is the actual structure of the plasma membrane? But we want to know, how do they actually get in and out? Well, we have two forms of transport. One's called passive, and the other's called active, and we'll talk about both, but we're going to start out with passive transport. Now, passive transport means there is no energy required. So the first type of passive transport is diffusion. It's movement of molecules from a high to a low concentration. And when molecules move from a high to a low concentration, they move down their concentration gradient. Anytime they move from one concentration to another concentration, that's called moving either up or down the concentration gradient. You move down the concentration gradient when you go from high to low, and you move up when you go from low to high. So in diffusion, those molecules are just spreading out, and the molecules are going from that really, really high to that really, really low. One way you could look at this at your house is you could get a cup of water and you could get a drop of food coloring, and you could just put that in, don't stir it, don't do anything, and let it sit on the table for an hour or so, and come back and observe it, and walk away, and then maybe in another hour observe it. And eventually, all the molecules will evenly distribute, and you'll have one color. It will be much different than when you first put that drop in. So another type of passive transport is called facilitated diffusion. Now this is still passive, meaning we don't require any energy. It is, though, the movement of molecules from high to low, just like before with diffusion, but we're using protein channels. Now this might be because our molecules are too large, they might be charged, they're not getting across that phospholipid bilayer. So each kind of molecule, though, is given its own special channel. So a water molecule cannot pass through the same channel that a glucose molecule passes through. They have to have a specific channel. For example, a water molecule has a specific channel called an aquaporin, and that's what kind of channel a water molecule uses for facilitated diffusion. A glucose molecule is going to use an absolutely different channel. So these proteins are made specific to the molecule. So one form of diffusion is called osmosis, and we use this a ton in biology. Diffusion is osmosis is the diffusion of water across a semi-permeable membrane, and our plasma membrane is a semi-permeable membrane. So we're using this all the time in cells. Water moves towards a higher concentration of solute until it reaches equilibrium. Now, equilibrium is just a state of equal concentrations, and molecules are still moving across the membrane, but concentrations remain constant. So water is always moving towards the higher concentration. So, for example, water is going to move towards salt. So think about this. When you eat something really salty, you want to drink a lot of water because you're really thirsty. Salt sucks the water out of your cells. So water is going to move towards the salt or the higher concentration because salt sucks. Water is constantly moving even if equilibrium has been reached. So once we reach that equilibrium, we're still moving across that membrane, but we're moving at an equal rate. We're constantly moving at that equal rate, and we don't have any net movement at all at this point. So in osmosis, we have different names for our solutions. We have hypotonic, isotonic, and hypertonic. Now, hypotonic is in a solution that surrounds a cell that has a lower solute concentration than the cell, and thus the water moves into the cell. So it's going to kind of blow up that cell. In an isotonic solution, the fluid surrounding the cell has the same solute concentration as the cell. So the water moves into and out of the cell at equal rates. Remember, we talked about equilibrium. This is the same thing. It's moving in and out at equal rates. And a hypertonic, the fluid surrounding the cell, has a higher solute concentration than the cell. Thus, the water moves out of the cell. Thus, it's going to just shrivel up, kind of. So let's look at a plant cell. In a plant cell, when you place it in a hypertonic environment, the higher concentration is on the outside. All your water is going to move out. And it's going to do something called plasmolysis. And your plasma membrane is going to move away from your cell wall. So this is our cell wall, and our plasma membrane is moving away. And it's just going to cause it to really wilt. Now, in a hypotonic, all that water is going to move into the cell, into the vacuole. And actually, this is what makes your lettuce really crisp, or you get that crunchy celery. Your plant cells actually really, really like this. In isotonic, this is kind of like your wilty lettuce or your flimsy celery. It's called flaccid, and it actually does not like this kind of environment. Now, as opposed to that, the animal cell loves the isotonic environment. It wants to be in the isotonic environment. The hypotonic environment for the animal cell, since it does not have a cell wall, it's going to blow it up. It's going to lyse. So it's actually going to hurt the animal. And the hypertonic is going to do the same thing. It's going to shrivel up. Okay, so we have one last type of transport. So we went through passive transport. We had diffusion, facilitated diffusion, and osmosis. Now we have active transport. Active transport requires energy in the form of ATP. So remember now that we have to transfer all of our energy into ATP. And some proteins are not just channels that open for facilitated diffusion, but they are also used as pumps to force ions across a membrane. When we transport an active transport, we're transporting molecules against their concentration gradient. So molecules are moving from low to high concentration, thus they are moving up their concentration gradient. Okay, so I hope you got lots of pictures, great notes, and we will talk about this in class.